Genes older than animal life itself were inserted into mice, and here is what happened. You may think that these two mice look alike, but the one on the left has darker eyes compared to the red eyes of the wild-type mouse on the right. With their little beady black eyes and dappled gray fur, the mice born in a recent lab experiment in Hong Kong are unlike any others of their species, or indeed any other animal. And yet, in spite of their fundamental differences, they could not appear more ordinary, a fact that reveals an astonishing truth about our evolutionary history. The mice were spliced with genes from a single-celled microbe called a canoflagellate. Though not an animal itself, the microorganism is closely related to them, having changed little since the time before complex multicellular life even existed. Remarkably, the success of a selection of the canoflagellate's genes is in some are as complex and multicellular as a mouse gives us new insights into the evolutionary origins of animal traits. Animal features that what is known as pluripotency, the ability for embryonic stem cells to differentiate and develop into the variety of tissues that make up a fully developed organism. In spite of not having this talent, canoflagellates have their own versions of the genes responsible for pluripotency in animals. By swapping mouse genes for the version found in canoflagellates, researchers could determine just how similar the two are in their functionality. By successfully creating a mouse using molecular tools derived from our single-celled relatives, we're witnessing an extraordinary continuity of function across nearly billions of years of evolution, says geneticist Alex de Mendoza of Queen Mary University in the UK. The study implies that key genes involved in stem cell formation might have originated far earlier than the stem cells themselves, perhaps helping pave the way for the multicellular life we see today. Pluripotency is thought to have emerged with the appearance of multicellular animals some 700 million years ago. 700 million years, that's almost a billion. So it stands to reason that transcription factors associated with stem cell pluripotency, such as those in the SOX and POU families, are thought to be restricted to multicellular animals. But prior research conducted on animal-adjacent microbes suggests that the origins of pluripotency predate multicellularity. If this is the case, it could be one of the drivers of animal evolution rather than the consequences of it. Canoflagellate SOX genes, have traits similar to those found in mammalian SOX2 genes. In mice, SOX2 interacts with POU member called OCT4, but canoflagellate POU genes are incapable of generating pluripotent stem cells. A team of researchers led by Ya Zhao and Daisy Lin Sena Tan of University of Hong Kong and Matthias Gierbid of the Max Planck Institute for Terrestrial Microbiology in Germany wanted to know what might happen if they replaced the mammalian SOX2 gene with the canoflagellate SOX gene. They grew cloned mouse stem cells and reprogrammed their genomes, replacing SOX2 with canoflagellate SOX, and these cells were injected into embryonic mouse blastocytes that were then implanted into pseudo-pregnant mouse surrogates to be gestated, birthed, and raised in a nurturing environment. The chimeric pups were born with a mix of traits based on their spliced heritage. Obviously, they were mice, but they had dark eyes and dark fur patches that indicated their mixed genetics. Otherwise, they were pretty normal, which suggests that canoflagellate SOX genes were able to create stem cells compatible with the mouse's development. This suggests that the tools for creating a pluripotency developed in canoflagellates before multicellular cellularity emerged. Canoflagellates don't have stem cells. They're single-celled organisms, but they have these genes likely to control basic cellular processes that multicellular animals probably later repurposed for building complex bodies. The findings suggest that the SOX transcription factors in chanoflagellate hundreds of millions of years ago were biochemically similar to the SOX genes that serve important functions in multicellular organisms today. The inability of chanoflagellate POU to produce pluripotent stem cells, on the other hand, suggests that POU members had to undergo modification to make up the role they play in pluripotency now. And these results could have implications for stem cell research and stem cell therapies, the researchers say, and they add an interesting layer of complexity to the story of how life diversified on Earth. The team writes in this paper, our data clearly shows that two of the main gene families involved in vertebrate pluripotency and key developmental genes across animals were already present before the origins of multicellularity. Eventually, their biochemical capabilities were excipated to build one of the defining cell types of a complex multicellular entity. And this was published in Nature Communications. It's on Science Alert by Michelle Starr. Please leave your comments. Thank you for your support. Please support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.